தமிழ் கோர்வை சேனல் சார்பாக அனைவருக்கும் வணக்கம். என் பெயர் கண்ணதாசன். Today I am going to present my video on Mendel's dihybrid cross. Just spend 10 minutes, students. You are able to understand the dihybrid cross very clearly, along with law of independent assortment, law of dominance, and law of segregation. Here I gave 3 links for technical terms 1 in genetics, technical terms 2 in genetics, and also for monohybrid cross. Please go through before dihybrid cross video, and also you can join the Telegram group and WhatsApp group so that you can get more screenshot and photographs from this video. Before we enter into this video, if you like this video, press the like button, share this video to your friends, comment on it, subscribe our channel, and press the bell icon to receive more such videos in future. Mendel's dihybrid cross. What is dihybrid cross? Dihybrid means studying of two characters simultaneously or trait simultaneously. Seed shape is one trait, cotyledon is another, cotyledon color is another trait. These two are not contrasting. Many of you thinking these are the contrasting character. No. Within seed shape there are two variation, round and wrinkled. These are the contrasting variation. The same way yellow and green are the contrasting variation within the cotyledon color. So here the two genes are involved. That is the reason why we call it as dihybrid cross. And dihybrid cross, here we are taking two pure breeding varieties. One pure breeding variety with round yellow seeds and another pure breeding variety with wrinkled green seeds. The genotypes are capital R, capital Y, small r, small y. And here you see the gametes carries only capital R, y, small y. This is because of meiosis. Being meiosis is the reduction division, right? Number of chromosomes are reduced. Number of chromosomes are reduced. Naturally, the number of alleles also reduced into half. When these two gametes are fertilized, a new heterozygous capital R, capital Y, small y heterozygous genotype is formed, in which or from which round and yellow seeds are produced. So the round and yellow variations are said to be the dominant variation. When the F1 generation form the gametes, you see there are 4 types of gametes are formed from anther side, and 4 types of gametes are formed on the ovary side. Okay, so how these 4 types of gametes are formed, that is what we are going to study in the next slide. You just going through this experiment. Okay, tossing a pair of coins simultaneously, what are the possible outcomes? First outcome is both coins may come with head, or first coin head, second coin tail, or first coin tail, second coin head, or both the coins may be tail. And here in the same way, this is the F1 generation cell. Okay, and it consists of a pair of chromosome. You see, pair of chromosome with a pair of alleles, capital R, capital Y, small y. Now, when this, say example, pollen mother cell undergo meiosis, definitely from this pair, only one can enter into a gamete. So when randomly these chromosomes move into the gametes, definitely you see the capital R having the chance of going with capital Y in one gamete. In another gamete, it is having the chance of going with small y also. In the same way, small r with capital Y and small r with small y. So this is the reason why Mendel put forward the law of independent assortment. Inheritance of an allele is independent of another allele. So capital R need not stay always with capital Y. It may also 50% gamete. It may go with small y also. And here the same thing. The true breeding homozygous crossing take place. F1 plants are produced with round yellow or dominant, and this is the anther mother cell, and this is the mother cell within the ovule. And when they are producing the gametes, four types of gametes are produced. You see, during meiosis, alleles randomly combine with other allele. Okay, capital R randomly combined with either capital Y or small y. So four possible combinations are formed in almost equal proportion. So always if you study along with the chromosome, it will be very easy for you to understand the symbols, right? Symbols through the inheritance from F1 to parent to F1, F1 to F2. So here capital R present in a pair of homologous chromosome in the same way capital Y, small r, small y. So in the gametes, only haploid number of chromosomes are present. You can understand here capital Y, small y in the haploid chromosome, and this is the chromosome present in the F1 generation with heterozygous nature. Okay, capital Y, small y. So the chromosomes are randomly moving through meiosis. When the F1 forming the gametes through meiosis, random movement occur. So four types of gametes on anther side and four types of gametes are formed on the ovary side. So in the F2 generation, you see, when these are the female you assume and this is the male you assume. So capital R, capital Y. So if it is 100, 25 may fertilize with capital R, capital Y, or 25 may fertilize with capital R, small y, 25 may fertilize with small r, capital Y, and the last 25 may fertilize the small r, small y. So this is the benefit of the Punnett square. In the Punnett square, we are able to see the gametes with their symbols on the male side as well as on the female side, and also how they are combining equally with other gametes. So the fertilization take place here. So we are getting the 16 combinations here, in which you can see the 9 of them are round yellow and 3 of them are round green and 3 of them are wrinkled yellow. The last one is wrinkled green. So you can easily remember it, students. You see, you remember the triangle first for the 9 round yellow. You see the triangle, and one inverted L for round green and another inverted L for wrinkled yellow. The last box for wrinkled green. So we are getting the ratio of 9 is to 3 is to 3 is to 1. Okay. So the result 9 with round yellow, 3 with round green. This is the new combination. 3 with wrinkled yellow. This is also new combination. 1. This is the wrinkled green. This is one of the parental combination. Okay. So in addition to the parental combination, we are getting the new combination. They are called as recombinations. The two recombinations are round green and wrinkled yellow. This is because of the law of independent assortment. First, let us discuss about the law of dominance. When we are bringing two homozygous individual in hybridization cross, okay, the character which appear in the F1 generation is called as dominant. The character which do not appear in the F1 is called as recessive character. And this is the law of segregation, otherwise called as law of purity of gametes. When a pair of contrasting factors or genes brought to the hybridization union, the two members, that is capital R and small r, may stay together, remain together without mixing. So you should not confuse here with the capital R and capital Y stay together without mixing. Not like that. The allelic pair remain together without mixing mean capital R and small r, the same way capital Y and small y. So they stay together. When the gametes are formed, they are separated. That's the reason why it is also called as law of purity of gametes, because the gamete carry only one allele for a trait. Law of independent assortment. Here the factor or gene capital R is independent in its inheritance, so that it may join with capital Y or small y. That is called as independent assortment. Right. So the inheritance of one gene is not dependent on another allele like capital Y or small y. So this is called as law of independent assortment. And here one more information about T.H. Morgan. He was the geneticist who was awarded with the Nobel Prize in 1993 for determining the role of chromosome in heredity. So that is the end of this video, student. Thank you very much for listening. Thank you.